Hello, everyone. My name is Antoine Hollebecque, and I'm a medical oncologist working at the Drug Development Department at Gustave Roussy in Villejuif, France. So on behalf of my co-authors, I would like to present data from the abstract titled Efficacy and Safety of pemigatinib in European patients with previously treated locally advanced or metastatic cholangiocarcinoma, a FIGHT 202 subgroup analysis. So cholangiocarcinoma is the most common primary malignancy of the bile duct, affecting 0.3 to 3.4 per 100,000 in North America and Europe. First-line treatment chemotherapy for locally advanced or metastatic cholangiocarcinoma is gemcitabine plus cisplatin. However, second-line chemotherapies have shown limited efficacy. For example, the median progression-free survival is between 2.6 and 3.2 months, and the median overall survival is between 6.2 and 7.2 months. The objective response rate is usually between 7.7 and 9%. Several actionable oncogenic alterations have been identified in cholangiocarcinoma, including alterations involving FGFR2. FGFR2 fusions or rearrangements are almost exclusively found in intrahepatic cholangiocarcinoma, occurring in 10 to 15% of patients with this cholangiocarcinoma subtype. Pemigatinib is a selective, potent oral inhibitor of FGFR1, 2, and 3. And in the primary analysis of 107 patients enrolled in FIGHT 202 with previously treated advanced or metastatic cholangiocarcinoma harboring FGFR2 fusions or rearrangements, treatment with pemigatinib monotherapy resulted in an objective response rate of 35.5%, a disease control rate of 82%, a median duration of response of 7.5 months, progression-free survival of 6.9 months, and an estimate overall survival of 21.2 months. So this poster reports results from a post-op subgroup analysis in 35 patients from Europe enrolled in FIGHT 202. So as shown in figure 1, patients eligible for enrollment into FIGHT 202 were adults with locally advanced or metastatic cholangiocarcinoma with documented FGFR status, progression after one prior therapy, an ECOG performance scale less than 2, and adequate hepatic and renal function. Cohort A comprised of 32 patients who had FGFR2 fusions or rearrangements, and cohort B comprised of 3 patients who had other FGFR genetic alterations. No patients were assigned to cohort 3 from the European population. Patients received pemigatinib 13.5 milligrams once daily, 21-day cycles, 2 weeks on, 1 week off. The primary endpoint was centrally confirmed complete response rate in cohort A. Secondary endpoints included objective response rate, duration of response, disease control rate, progression-free survival, and overall survival in all cohorts. Efficacy was assessed in all patients who had centrally confirmed FGFR status who received at least 1 dose of pemigatinib. Safety was assessed in all patients who received at least 1 dose of pemigatinib. So the data cutoff used for this analysis was identical to that of the published primary analysis. So as shown in figure 2, at data cutoff, 25 patients discontinued treatment, 21 due to disease progressions, 10 patients were continuing treatment. Patients characteristic of the European population shown in table 1 were generally consistent with the FIGHT 202 total study population. The age of patients who had FGFR2 fusions or rearrangements range from 34 to 77 years, with a median age of 57 years. 34% were men. Majority of patients were from France, and 31 out of 32 patients were diagnosed with intrahepatic cholangiocarcinoma. So the age of patients who had other FGFR2 genetic alterations range from 67 to 70 years, with a median age of 67 years. 33% were men, and all the patients were from Spain. And 2 out of 3 patients were diagnosed with intrahepatic cholangiocarcinoma. So as shown in figure 3, of the 32 patients with FGFR2 fusions or rearrangements, 23 different FGFR2 partner genes were identified, 21 of which were unique to individual patients. So the most common FGFR2 partner was BIC1. It was the same in the global cohort. As shown in table 2, in patients with FGFR2 fusions, the centrally confirmed objective response rate was 40.6%, with 1 complete response and 12 partial responses. The median duration of response was 7.5 months, and the disease control rate was 87.5%. In patients with other FGFR genetic alteration, none achieve response. Objective response rate of the European populations were generally consistent with the published findings in the FIGHT-202 total study population, whereby 35.5% and 0% objective response rate was achieved in those from the total study populations with FGFR2 fusions and other FGFR genetic alterations, respectively. Most patients had reductions in centrally assessed best percentage change from baseline in target lesion size, as seen in figure 4. So as you can see in figure 5, the objective response rate in patients with FGFR2 fusions was similar across demographic and disease subgroups. Kaplan-Meier estimate of progression-free survival and overall survival are shown in figure 6. The median progression-free survival in patients with FGFR2 fusions or rearrangements was 6.9 months and was generally consistent with the published findings in the FIGHT-202 total study populations, which was also 6.9 months. The median overall survival was 14.7 months. However, overall survival was not mature at the data cutoff. So as seen in table 3, the most common adverse event in patients with FGFR fusions were diarrhea, 80%, alopecia, 66%, hyperphosphatemia, 60%, and nail toxicity, 60%. Adverse events led to dose reduction in 20% of patients due to either stomatitis, palmar plantar erythrodysesthesia syndrome, or asthenia. 57% of patients experienced dose interruptions due to adverse events from either stomatitis, palmar plantar erythrodysesthesia, pyrexia, fatigue, or asthenia. Adverse event did not lead to any treatment discontinuation. Among the clinically notable adverse events, hyperphosphatemia occurred in 60% of patients and were all of grade 1 or 2 severity. So these events were managed with a low phosphate diet, phosphate binders, or dose reduction or interruption. Hypophosphatemia occurred in 14% of patients and was the most common grade 3 adverse event, and it occurred in 9% of patients. None of these events were clinically significant or serious, and none led to discontinuation or dose reduction. One patient experienced a serous retinal detachment event of grade 1 severity, which did not result in clinical sequelae. Adverse events reported in the European populations were generally consistent with the published finding in the FIGHT 202 total study population. However, 9% of patients in the total study population discontinued treatment due to adverse events. So in conclusion, the efficacy and tolerability of pemigatinib in the European population of patients with locally advanced or metastatic cholangiocarcinoma harboring FGFR alterations were similar to the published findings in the total study population. Adverse events were manageable and consistent with the mechanism of action of pemigatinib. Pemigatinib treatment resulted in an objective response rate of 40.6% and a median progression-free survival of 6.9 months in the European cohort. 23 unique FGFR2 fusion partners were observed, supporting the use of fusion partner agnostic testing. So the study demonstrates the potential therapeutic benefit of pemigatinib for patients with previously treated locally advanced or metastatic cholangiocarcinoma and FGFR2 fusions or rearrangements. So a phase 3 study is ongoing in the first-line setting to evaluate pemigatinib versus gemcitabine plus cisplatin in patients with cholangiocarcinoma and FGFR2 fusions or rearrangements. Thank you very much for your attention.